Okay, hello everyone. Great to see so many people in the second webinar today. I'd like to introduce Dr. Martin Schobben. Martin is a postdoc at Utrecht University in the Netherlands. And as a paleobiologist, he combines paleontological, sedimentological, and geochemical methods with quantitative approaches. His research focuses on the evolution of life in conjunction with the Earth system throughout geological history, such as the great mass extinctions. Last year, for example, he was invited to the Dutch National Radio to talk about his research. Martin has a keen interest in all aspects related to data collecting, analyzing, and visualizing, and he uses primarily R programming for all of this. Pages ECN has also organized another webinar on R in 2019 when Simon Goring explained the package R Markdown. This webinar is on our YouTube channel. This webinar today will also be eventually uploaded to our YouTube channel, but this might take a few days. So it is recorded just so you know. Yeah, today Martin will show us his best practices in data exploration and data visualization, more in the form of a workshop, and it will last roughly about 1.5 hours. And the idea is Martin has prepared 4 blogs, and he will first explain his steps in some slides, and then switch to R and show how to actually do these things. If you have questions in the meantime, type them in the chat, and we will try to answer them in these interactive periods when he works in R. Also, please mute yourself, and then I think we can start, and I hand over to Martin.
Great, thank you, Lena. I'm going to share my screen. Let's have a look. There we are. Make it a full screen. Yeah, thank you very much, and thank you for joining this webinar. I'm first going to briefly give a little bit of an introduction of my career and how I became more involved with the data aspect of the earth science part that I do. And for that purpose, I'll show you now this Venn diagram. And maybe you've seen this before. We as scientists, as earth scientists, would normally be educated and trained within this overlapping area here of traditional research, which like intersects our knowledge of math and statistics, and on the other hand, our expert knowledge, so our expert knowledge of the earth system. And throughout my career, I focused more on the aspect of data and also the usage of computer science and computer technology mainly. So I'm talking really about programming and R, as you will see in a bit, and it's of course also the title of this webinar. And so I shifted slowly, gradually more to this intersection here where we have then the data science. As many of the curriculums, at least in my experience of universities, don't have any formal training, or there seems to be a lack of training in especially this computer science part and the technology usage, that I want to give you little pointers to what would be the best practices, at least in my experience. It's more about things that I found out throughout my PhD and postdoc that I think that is very useful to know actually already at an early stage when you begin a project. And as Lena already explained a little, I divided the webinar into 4 subsections. So first I'm going to talk a little about the data geoscience project as a whole. Then I talk about the exploration of your data, the visualization mainly. I talk about modeling, modeling in the sense of extracting the really important signals from your maybe very noisy data. And as a last part that is maybe a little bit counterintuitive, so I wrote it out a little bit more extensively here, is the wrangling part of the data science or the data exploration, which is the loading, so the importing of your data into R, tidying it, and also then transforming it, so creating new variables or renaming them, so that we first can right away start with the exploration of the data, and then only later on I'll show you how to get to the right data format. But normally in your project, this would encompass quite a lot of time, section 4. And so I aim for 5 to 10 minutes of introduction every time, and then 5 to 10 minutes of live coding and showing you stuff within RStudio and R. And also have some time for questions then from your side. For the purpose of this webinar, I created the Pages R package. My experience from this morning is that people could install it. I hope you succeeded as well. It is maybe worthwhile to install it again if you installed it yesterday, because I made some little updates in the morning, especially to the slides. So if you really want to look at the slides and compare them to what I tell you now. And then the web page and the GitHub repo, I also put the links in the chat of this webinar, so you can easily find that. So, and then you load pages with library as you would do with other packages. Yeah, that's that. And so I constructed this course with the R for Data Science book really as the fundament of everything. So this is the book by Hadley Wickham and Garrett Grulemund, which is really a groundbreaking work. It's really a fundament of data science with R. But what I did, instead of the examples that they use in the book and also the example data sets, I include my own data sets from geology, mainly stratigraphic examples, which you can then load with the package. Now, this book revolves completely around a set of packages which are called the Tidyverse collection or the Tidyverse universe. And they say that these packages are opinionated, and that is in the sense that there is a certain philosophy about how data should look like, how a tidy data frame should look like. And it gives you also quite informative messages and warnings when you do operations that are maybe not to the greatest benefit of the data science project or the transformation that you're about to do. For instance, within the ggplot that I will show you, it will often complain if there are NAs. It's not really complaining, but it tells you that they are there. So you shouldn't worry about that, but it's more about informing the user of what's really going on with your data. Then I said already, I talked already a little bit about ggplot now, but this is really a very important package nowadays in R and very popular as well. So many users, and this is a modular graphing device. So you can actually mix and match many elements and come to a unique solution for the problem that you have with your data. And I will explain quite a lot of details about this package in some minutes. Then I will also introduce some functions of the dplayer package. This is really the manipulation, so the transformation, renaming, reordering, and creating new variables. The reader package, so for importing your data sets. And lastly, also the tidyr package to cast your data into the tidy format that are used by the other packages again. But as I said before, so I included my own data sets now, and data from my own past, so projects that I worked in, and it's about stratigraphy. So we looked really at variations in the value of a variable through time, essentially, or it's at least an ordered sequence. And so this time unit can be then measured in height or depth, depending if you're working on sections or cores. And it can sometimes then also be calibrated to absolute time if you have an H model. So as I said before, if you have in the R package pages, if that works for you, You have the lazy load data sets, Bonneborg and Kujor. So if you really type only the name and press enter, you will print then this data frame. And also if you use the question mark in combination of the data set, you will get some information about what the variables are and what the units are of the variables. And these two sections that are included are both Triassic Jurassic boundary sections, so 200 million years ago. And within the study that I used them, I essentially looked at the total organic carbon and the carbon isotope composition of that. And I'll show you also already here a picture of the Bonenberg site in Germany, where you can see a clear distinction of three lithological units. So the black and then the red and the yellow, which is here would be the boundary from the Triassic to the Jurassic, which will come back later in the talk and webinar. Now, to start off, to wrap everything together, I'm going to talk about the Data Geoscience project. So in a minute, I will talk about all these aspects of understanding your data and importing your data, and the combination, which is then the programming aspect in R. But I will now talk about the whole project around that and how you can best set up a project in my experience. And for that, I would always suggest to use the R Studio project. That is because when you are a beginner using R, you are very interested in the output of R. So you do a statistics analysis on your data, you get an output and you use those values, or you create a figure and it's to the standards that you want. And so you save it as a PNG or a PDF or whatever format. And that's what you want to do. But when you grow and become more of an expert in R, the emphasis shifts a little bit to the scripts and functions. You are more interested in which script belongs to which figure. So how can I recreate a figure that I made in the past? And so then you really start to work with projects, because then you combine everything together in a neat package. And the RStudio project is the solution for that. And besides that, it also gives you the benefit if you save something that the pathway always points then to this directory. So you don't lose a figure somewhere in your system and you have to search every folder where you now actually put the figure that you printed or saved to a file. And then in some minutes, I will show you then the sequence of windows that you will see when you set up a project, and so that you get started with a project, with an RStudio project. Yeah, and if you then set up this RStudio project, my suggestion is that it would be wise to have a clear directory and file structure. So where your data is, and also metadata describing the data. And that could look like something like this, like I often use. So you have a data raw directory within this project, which contains really the output of the machine or your count data. If you do paleontology, for instance, you have a data folder where you have your process files. So if you do already the tidying and the formatting of your data, then you put the output in this folder. And you could use an R directory for containing all the scripts. This file that you see here would be then essentially your project file that sets up this RStudio project. What is also wise to do is to make the raw data files read-only, so that you only read it in with R and that you don't accidentally delete an observation or mess about with the data that is in these files, because they are just so very important. I would also suggest to keep always backups of at least the data raw, but I would also keep a backup of the whole project essentially. And then in the end, if there is an outcome from this project, you could then publish all these aspects along with your paper. And as I know now that there was also a webinar, I skipped your point. So the R scripts, I would always advise also to have a clear documentation of all steps. And then if you are able to use R Markdown, and you had apparently a couple of years ago a webinar in R Markdown, that would be very useful to use that as well to describe all the steps of the data analysis that you do in R. This is one aspect that I want to introduce early on already. Not everybody might be familiar with the pipe function. This is mainly a sort of a visual or a clarification tool within your code. For instance, when you start coding, you often use intermediate steps, intermediate objects to save the output of a function. So for instance, if you do a grouping of a data frame, you would save that as an intermediate, and then you do another operation with summarize here with getting the mean of your count, and you save that again. That is one way of writing down your code. But the problem with that is that you get quite a lot of clutter in your environment. And you also have to be very creative with all the naming of the objects for the intermediate steps. And then sometimes you get object names that completely spiral out of control. They are very long and complex to remember. So some people use as a solution to that the wrapping of multiple functions on top of each other. So here you would have then the grouping within the summarize function call, and then you have right away an output. But the problem with reading this sort of code is that you have to sort of read it from the inside to the outside, because this will be performed first and then this function second. So the pipe is the solution to that kind of problem, which is really a writing style. You should call it a writing style for your code. So then you can do the group by and then pipe it into the next function. And so what that does essentially is the output, the data frame that comes out of this function call is put on the first argument of the next call. And then you don't have any intermediate steps, and that's right away saved in this object sum here. And you will see the pipe return in some of the examples later on. So you know already a little bit about what it does. It's a really useful tool, so I would look into it. So now I will break off the slides and show you quickly how to set up the project in RStudio. Let me see. So if you set up, I hope most of you will have RStudio. It's a really convenient tool, as I said already before. So if you want to set up a project, you go to the upper left corner here and you click on File. And instead of making a new file, you do a new project. And then you start this sequence of windows. So you start off first with selecting a directory, creating a new directory. You would select the new project in this case, and then you would name the project as you like and save it at a location that you like. And there are also other options here that you can click, like version control, but that's maybe more for the experienced user. But I would also suggest to look into that kind of details later on. And for some more information about this, I also created, so if you load the pages package that I developed for the webinar, you will find in a vignette. So vignettes are a long format documentation of a package. You will find under vignette project a more detailed description of what I just told you. So setting up the RStudio project, the file structure as I would suggest it, and also a little bit about version control, but it's more the experienced user then. So Lena, we would have time for some questions. Are there any questions?
I don't see anything popping up yet, but maybe people need some encouragement, but so far, no questions.
step of water first. And then I can maybe just continue then.
Yeah.
You would say?
Yeah. I mean, yeah, people can ask questions. So whenever you feel like you have a question, just write it in the chat anytime.
Yeah. And I can also go back in the slides to previous section, doesn't matter. Oh, that was wrong. So then we can start now with the data exploration part. And we are going to look now a little bit more into detail to this graphic that I showed before. That is also from this book that I introduced before, and also in the introductory email. So R for data science. And we're going to look at the exploration now. So in this blue box here. And we're going to look at the transformation of data and mainly actually the visualization. And for that, we're going to use ggplots. I guess many of you have heard about this device already. And this quote of John Tukey actually says everything already, why it's so important to visualize data. So the simple graph has brought more information to the data analyst's mind than any other device. And so before doing any modeling or other type of analysis, you would always first start by visualizing your data. And ggplot really helps with that. A ggplot is based on the grammar of graphics of Wilkinson. And so it is the interpretation by Hadley Wickham, also the writer of the book that I introduced before. And it is meant as a sort of a coherent system for describing and building graphs. And here I show you some of the layered components, so these modular components that I talked about before of ggplot that use then this grammar of graphics. So first of all, I have to explain that in this representation, the things that are wrapped or bracketed by these arrows and are written in uppercase can be changed. So this would be your own data set then. So first of all, we start off a ggplot by just ggplot call. That's really the basis. You enter your data, so your whole data frame enters here. And then you can add the modules to that. So you do that by using a plus sign. And the most basic plot uses then a geom function. And a geom function is a geometric object. So it visualizes the data. And there are many types of geoms that you can use. And I will show you a couple of them in the next slide. The geom function needs a mapping. That's one of the most important aspects. A mapping is a mapping of your data to a visual attribute of this geometric object. And I will show you in a minute what that really means. So this is really this grammar of graphics. And I will also show you another couple of arguments of the geom function, which is stat for transformation of your data. You can actually do statistical transformations already with the geom call. The position and the orientation that will come back in the exercise that I have at the end of this section. And then also the facet function, which is a subsetting of your data. Now, let's start off with a very basic plot. So the scatter plot, or in our discipline, we also often call it a cross plot, maps down each observation as a point, and then it maps it on a horizontal and a vertical position by using then the X and the Y coordinates on your Cartesian coordinates. So, and I haven't said that before. So every example that I show you here now, where there's actually data inserted in the functions, you can reproduce with the package. So you can just copy paste from the slides if you have the slides in front of you. So if you enter then the Boneberg data set and you do a mapping call and you put in there the delta 13C, that was the emphasis of my study, on the x-axis, and the height to the y argument, and you get just your delta 13C versus height. So you get the variability of this value of this variable over time, essentially. So this is sort of a first step in of the data analysis, what you can do with these proxies. We often have in mind, for instance, for delta 13C, that it would mean that we see a change in the carbon cycle. But it doesn't stop here. So this is just a very basic plot. And now let's move on to adding some more details to this plot. So as I showed before, the data set that I use has a distinct subsetting or subdivision of lithological units, 3 of them. And what I will do now in the next representation and the next function call is map also color. So I map the stratigraphy to the color of the data points. So this is another visual attribute of your geometric object where you can assign a value to. And that doesn't have to be only color, it can also be the shape of the point. And if you use a line, you can also do, for instance, line type. So there are many aesthetics that you could essentially change. And here I use then the stratigraphy, which is then a categorical variable. And that gives already some more information. And it also shows one of the strengths of ggplot is that it automatically generates this legend here. So you can actually match then the color also with something that we understand, really the data. So we know that, for instance, these points are the 3 latest beds, and the selenoid and tone formation would be up here. And we also see quite an interesting pattern emerging already here, that there seems to be distinct clustering in the data. So it seems that also lithology has maybe an influence on the values of the delta 13C here. So that's data analysis as a starters. But we can make it more interesting, more complex. We can also do statistical transformations of the data. It just does that internally within a ggplot call. And I'm going to show you now in the next slides the usage of the stat argument and the facet function. And first, I will start with statistics then. So often when we do data analysis, we want to visualize the distribution of our variable and the values of this variable. And we ask ourselves a couple of questions. For instance, which values are common, which values are rare, which are outliers in our data, and can we also sort of discern patterns in this distribution. So if we look at this figure that you can find back in the book, then the actual values would be plotted like this. Doesn't give a lot of information, but if we would make a histogram out of that, and this is a rotated histogram, then you can also already see the frequency within a bin here. So this is bin data of a certain value. And there is also another way of representing this, and that is the box whisker plot. For those that don't know, the idea behind that is that we have then a median, so the 50 percentile drawn. We also have the interquartile range drawn, which is the box of the box whisker. And then we have also outliers drawn, which are 1.5 times the distance of the interquartile range then. And we have the whiskers that extend to the last point before the first outlier. And I'm going to show you now an example where I use that for the isotope data as I showed before. And essentially, I'm going to compare then a categorical variable with a continuous one. So we go back to the Boneburg example. So it's included again in the ggplot call. And then I'm going to use now a different geom. So we have very simple for boxplot, the call geom boxplot. Now, for the mapping here now, we use then, as I said before already, for the Y, the categorical variable, which is then stratigraphy, which are these different lithological units that we saw before. And then the statistics are performed then on the subsets of the data of the delta 13C. And this transformation of the data is done internally by this call to status boxplot. But this is actually the default setting of geom-boxplot, so we actually don't have to provide that. So I'm going to show you that now. So you don't actually have to include this. And in this plot, I also did another tweak added to the plot, because actually the stratigraphy is also ordered categorical variable. So I ordered, or I reordered the stratigraphy according to height again. So this is the correct way of displaying it in a stratigraphic sense again. And then we see that we really have distinct differences in this isotopic value of the different units, where these might be more similar and this one unit is far off to the more positive side then. And also you see that there are differences in the variability overall, where there's low variability here and a lot of variability there. So this is a beginning of data analysis that you could do in the project. Now I will move on to another aspect that I find very helpful when performing multi-proxy studies. So when you have, for instance, a lot of geochemical measurements. And for this function call, so faceting splits then the data according to a categorical variable again, but it splits it in different panels then, so that you can see the subsets of the data in detail. And within this call, I included Bonneburg in a different format. It's a long format data frame. I'm not yet going to explain what a long format data frame is. But it is a very useful tool in this kind of multi-proxy studies. And now I went back to the geom point geometric object and mapped in the value and the height again, the same as I did in the initial scatter plot. And now you see that the boxes above here, those record the different types of measurements that are done. So calcium carbonate, isotopic composition. This is potassium over aluminium ratios, sodium over aluminium, total nitrogen, and total organic carbon. If you do use the pages package and you do a question mark Bonneburg call, then you can actually see more information about this data set. And then the facet grid itself does this subsetting. So the measurement, so the categorical variable is doing this subsetting of this data. I used here also this argument scales and then set it to 3x with underscore then to better visualize the ranges for this X scale, which are quite different for all these different subsets. So I find it a very useful asset for a first initial exploration, and then you can actually already start looking at covariation, for instance, between different proxies. So now I break off to the next section, and I'm gonna do some
You want
live.
some questions in between, or first show?
I'll first show the example then, and then because it's also just ggplot then, and then questions can be afterwards.
Okay.
So for this, I'm gonna use a different data set. I'm gonna use the Long data set, which is a paleontology data set. And you get some more information if you do this call. So we have 180 observations. Then we have again height in a unit centimeter. We have a type, which is a characteristic of the paleomorph assemblage. And then we have a label, which is type of paleomorph assemblage and the actual count. So how many occurrences were there under the microscope for this particular subset. And what I'm going to do now, I'm going to make a plot where I do an area plot to show compositional changes within the paleomorph assemblages. And for that, we start off with the ggplot then. I do very explicit writing of all the arguments, which is not always necessary, but I think for the purpose of this webinar, it's quite good to include that so that you see where everything belongs. And so now I'm going to use a different call. It's a geom area. And we're going to define the mapping here. And we need then the AES function, so the aesthetics. And for X, I'm going to use now the count. And for Y, I'm going to use height. And then I'm going to fill this area with a color to make it visible, otherwise you wouldn't see it. And I do that with a label. For this types of plots, it's very important to add also the orientation, because a ggplot would normally draw lines in a different direction. So in the X direction, X-axis direction instead of the Y-axis. And then we have another categorical variable that I'm going to add as a facet grid here, which is again the columns where I assign a variable, which is in this case the type here in this categorical variable. And I leave for the rest everything as default for the geometric object. And then we get a plot like this. So these are really total counts of, for instance, then the terrestrial and aquatic elements and the spores and pollen, which is maybe not very useful at first in an initial analysis, because we really want to see how the closed sum, the fractional changes, the relative changes are in this assemblage. So I'm going to tweak now the geom area call to enforce also a transformation on top of this data. And for that, I'm going to use the precision as I already promised before. And we're going to change that from the default stack to fill. And then we get a plot that is maybe already a bit more interpretable, where we see then, for instance, the changes in aquatic and terrestrial input, and differences in spores and pollen contribution through time. So we see actually better actual changes in the assemblage. And to give you an idea of the transformation that is already done for you within the data, I'm going to show you the transformation outside of the data using dplayr, and see that it's actually quite a lot of lines of code that you don't have to include now, because otherwise you would have to do for yourself a group by, so grouping of the data set, where we would then use height and type. If you do a grouping, you will see that also in your table or your data frame on top here. And then you have to do the mutation, so the creation or the recasting of this variable yourself, so of the count, by taking the fractional abundance of counts of these grouped. Oh, sorry, this is a mistake. So it should be the sum, the total sum of this group. So this would be this transformation. If we add that then to the ggplot call, I'll leave now this empty, because the first argument gets via the pipe now the data frame. And we add the mapping to retype now everything. X is count and Y is height, and fill is label. Don't forget the orientation, which is very important. And we also do the facet grid call, where we use columns again, the variables, and then include type. So again, this part. And we see we get the same plot, but now it's all outside of the function call. So you really save a lot of typing there. For this part of the seminar, you can find the information in the vignette Explore if you want to get an idea of or redo what I've done now before. Yeah, Lena, let's go to some questions then.
The first question was for your first cross plot that you color code. The data has at least 3 headers then. So height, your delta 13C, and the stratigraphy, if that's right. So that all. Yeah.
So it's not only bivariate then, it's multivariate in the sense that you also add a third variable there, which is then the color. So with aesthetics, you are not only tied to only your X and Y coordinate for displaying variables, you can actually include more. And so you could actually include even more than just color. You could also change the shape, for instance. Yeah.
All right. Another question was, there are 2 visualization libraries that I find confusing. And so what is the difference between ggplot and ggbiplot?
ggbiplot, I actually don't know. I guess ggbiplot is probably, so there are a lot of add-ons built on top of the ggplot package. For instance, ggalley for crossing of data. Let me think of other ones. ggrepel for labeling of data. So I guess it's just an add-on to ggplot, considering that it has gg grammar of graphics included in it. So ggplot2, I'm going to type it in the chat. ggplot2 is would be the package that I'm talking about.
All right. And another comment or question is more how to format and organize your data so that you can do all these.
I'll get back to that in the 4th section of this webinar. So please hold on.
And another question is, what is the function of ACE?
Yeah, it provides the aesthetics. So it's a function that defines the aesthetics. We can actually just look at the actual definition here. So always use the manuals, very important. So it describes how the variables in the data are mapped to the visual properties. So this is really this grammar of graphics. So it maps the visual properties to the geometric object. So a geom is a geometric object. A geometric object is, for instance, the point or the line or the area that I showed. I hope that clarifies. With 2D box plot. Because it would already be bivariate as a categorical. Oh, okay. You mean a grouping within the box plot? Okay, that's yeah. Could try to do that.
Of course, we don't know the level of expertise of R with all of the participants. Of this is the webinar is called First Steps in Exploring Data, but feel free to ask any questions you have for Martin, as simple or as complicated as they are. And then Martin can decide whether to answer them during this webinar or maybe later.
I'm also first going to share the website once more in the chat and the GitHub repo. Let me think about so what you mean with Bohnenbusch. Gym box plot. Um, okay, this was my original. Maybe. Oh. I guess you mean like this. So if you would have another categorical value, which I don't have at the moment, or do I? Then you can actually have also the second categorical variable you can include as fill then, as the fill aesthetic. Let's move on to the next section. And so I'm going to talk now a little bit about patterns and models. It's quite a brief section, I would say. It's more about like discerning, extracting the patterns and the signals from your data. And it is not really a confirmation or really a formal inference test. So we're not gonna pose a hypothesis here and want to get a formal answer out of that. It's more of extracting more information out of your data, and then as visualized also in this graphic, it puts you in this loop where you then model this pattern, and you have maybe an intermediate hypothesis, and you see whether you see that also if you model your data and visualize your data. If not, you start over again and take another approach, and that would be really then the exploratory data analysis approach. And so for my data, so the Bonneberg geochemical data set, I'm looking here at covariation of two continuous variables in this case. See if we see some covariation, some correlation in these subsets of the data. This is again a little different data set. It's again a long format, but I call it here cross because I crossed it in such a way that you can look at the isotope composition against the different other continuous parameters or variables within the data set. So calcium carbonate, height again, we have seen that already actually, potassium of aluminium, and the other ones that I already mentioned before. And this is just a sort of a generic function call. And it uses again then this facet. I use a facet rep here, which is a variant of the facet grid that I showed before, but it is 1D instead of the 2D before. So the grid is really a matrix, and the rep, actually the name says it already, wraps it in a sort of a rectangular format. I still use also the color then for the points for the stratigraphic units. And then we can see that there are maybe already some patterns emerging there, that you see that the high isotopic values correlate with low potassium of aluminium, and the lighter, the more negative values of the carbon isotope composition with higher potassium aluminium, but maybe more expressed as even this TUC to delta 13C correlation, which seems already, if I would see data like this, it's something like exponential, not really linear. So let's look into that into more detail a little bit. And for that, I'm going to use now, for the modeling part, a new geom, and this is the geom smooth, which introduces a model here. And I'll leave everything to default here now. And what is also important to observe here is that I included now, and that is different from the other function calls that I showed before, I included part of the aesthetics mapping in the global ggplot call. So this value and the delta 30C I put here is a global mapping. And then for the points, I add a local mapping associated only with this layer of plotting. So only with the points I associated stratigraphy. For geom smooth, it uses essentially then the global aesthetics mapping. So the global data set. So what it does here, it puts a model on top of this data for the global data set. And so we see then also what we've seen already in the last plot, just by looking at the points, is that we have indeed a trend here going down. And for TUC, you see the same. And I have to say for the default statistic, for the default modeling method of geom smooth, it's a locally weighted regression, Lewis fit, which is then a non-parametric approach. And now I'm going to move to a more in detail visualization where I really look into detail to this TUC, to delta 13C, and use the more well-known linear model for representing this this trend. So I'm going to fit a linear model here, and that is done by adding then to the geom smooth call a different method, and you put here then this character string LM, and LM might be familiar to you who uses R already a bit more, because LM is your standard base linear model call, where the base call would look like this. You have your response variable, so your Y, and you have then the tilde, which is the formula expression, and then your TSC is the predictor of this model. And then Borneberg would be the data. So that's the same what is done here in this geom smooth call. It's added as a new layer, as a new geometrical layer to the plot. Not really content yet with how that fits to the data. It's not good at all, I would say. What I do now in this next call is a transformation on top of TUC. I do it within this aesthetics call here, in this mapping, a log transformation. We see that we got quite a bit better call. So there seems to be some sort of an exponential relation there between the two. I would suggest not to do too much of transformations within the aesthetics call, and there is actually a more elegant way of doing this by taking a different scaling, but it's a little bit outside of the scope of this webinar. So for now, I would say this is an option. So then if we do some ad hoc explanation of our data, then it seems that, well, maybe delta 13C in this site is not reflecting really the carbon cycle. Maybe it's also a consequence of other chemical properties of the stratigraphy that we are looking at, and maybe also the stratigraphic units one, because we see still this clustering of these units. So this is really just the beginning of what could be done with models, just by using only ggplot, but you can and maybe should also do more detailed analysis, more complex models. So for instance, you want to also use maybe multivariate regression. So in the case with this categorical stratigraphic variable and the TUC, which is continuous, you might actually combine that in one model. And then you have maybe additive components and also interactions. So how does one variable interact with another and has, again, an influence on the response in one or the other way. We also should always diagnose whether a model fits our data. So do the residual diagnostics. Look also at correlation structure, because time series have often this correlation between time steps, and grouping structures, as seen with the stratigraphic, with the lithological units here, might be important here as well. So maybe mixed effects models would be a way forward in your analysis as well. I can't explain all of that. That's beyond the scope. Also not my expertise in that great detail, but I would suggest this as further reading, or at least that helps me quite a lot. This book I already had included in the introductory email, but then you also have this book from John Fox and Sanford Weisberg, and then Alan Zuur has a book on mixed effects models, and all these books include examples of R and also the associated packages with that. So if you have the Pages package installed and you render my slides, you can actually find the references. And they are also on the README file of the GitHub repo that I supplied in the chat. So let's break off again to the RStudio console. What I want to show now is a nice example why we should always visualize our data first and then use a model on our data. And I do this by using the data source dozen dataset. Maybe some of you know it. It's already included in the package. So if you do this call, you will get this data frame with what is now 1846 observations. And we have this dataset categorical variable, which is a subset of all the data sets that is included there. Oh, you see here the different data sets included here. And that's just an X and a Y variable. What would happen now if we just start introducing a model to this data set, a linear model in this case I want to show. Keep doing the very explicit function building here. And we use the GM Smooth that I showed before. And I also use then this linear model as in the last slide. And we need then also to use again a facet rep because we have these different data sets and we want to use and display the different subsets then. Use vars to supply this variable. Oh. And I forgot now the most important part of the function call, the mapping. I did that now 2 times. I did it in the morning and I did it now. And the y is y, so x is x and y is y. And then we get this whole raster of plots. And what do we see? We see models that have the same slope and the same intercept. So you could argue if you see that you might be dealing with quite similar data sets. But there comes the big surprise with this data. Oh. So if we instead now just plot the points, we see that we have been misled. And we see also the data source cropping up there. Move this away. So we see that we have quite a variable data set here. It's completely different, but they all have the same variance. They have all the same mean. So if you plot also a linear model to that, you get the same kind of trends. And this type of plot is called an Anscombe plot. It's named after a famous statistician from the last century that first showed this important aspect of visualization before analyzing data. Again, more information is included in a vignette model and also on the website if you do this call, and you see some more information on the data source. I also included some colors here. And that's it for this section. So Lena, are there questions?
There are 2 questions. One is, do you need to define the LM function for your data before calling it in ggplot?
No, it does that automatically. And you saw also for the last call here with the data source that the formula used for this linear model was now this kind. You can also modify it by, for instance, excluding the intercept. It doesn't make sense the printing what I did now, but it functions the same as just the base LM function. So you can actually also add these other arguments that you add to the LM function, but essentially you don't have to define anything. You can use all the defaults if you want to. And so the transformations are all done in the ggplot call.
All right. Another question is, how do I control or modify the width of the smoothing?
The width or the what?
The width, yes.
In what way?
I do not know.
It's hard to interpret this question.
I guess the I mean, maybe Rodrigo, you can ask the question yourself if you want to, instead of me interpreting.
Yeah, hello.
Oh, okay. For the lowest fit.
Yeah. For example, you have a lot of points in you.
Oh, you have a bit of feedback there.
I have a friend here who is listening the talk also, but there is a problem with the audio.
But you can define the width, or it is actually a lowest model, so you can define a span. So that is a nearest neighbor span. So you can see it here. And you can also include that into your GM-smooth call. What is also interesting is maybe instead of using Lewis as your method, using a GAM, so MGCV, so generalized additive models, which controls, has an sort of an automated control on the smoothness of your regression line. So if you want to have a smooth regression line. And that can all be included in the GM-smooth as well. Was there anything more, Lena?
There's one other question, if you can recommend any literature that is very specific for paleo data in R.
With R, huh, that's a good one. Not really. So for paleo data, so the Zuur book that I showed before on this slide, at least it's adapted to ecology. So that's already getting a little bit closer to what we do, but really paleo data, I wouldn't know. For the statistics, I would always suggest, I have to check that now. Would be Hammer and Harper, but I don't think they have examples in R. But that's just statistics for paleontological data. So
maybe related to that, there's one other question. If you have any advice for working with the time series and type series objects, packages, instead of using data frames with ggplot.
What was, yeah.
And can you repeat the literature you just said?
Yeah, so the literature.
Maybe you can type it in the chat.
Yeah. So if you go to the GitHub repo, oh, I have it actually open here. Put it in, and you just look at the README at the bottom. I have the literature added to it. For the time series, I forgot the name of the package. There is a tidyverse package for time series, and I would also suggest to look at tidy models. This is a collection of tidyverse packages essentially made for modeling. So we can have a look at the manual for that. Oh, maybe I don't have it installed. Yeah, I think this would be a start. So if you want to continue with philosophy of and the grammar of tidyverse, then tidy models would be the package, and that must also have some ways of dealing with time series. All right.
Yes, I don't see any more questions right now.
Okay, then I go to the last part. And now we get to the part where we actually get our data imported and tidy it and also transform it. So we move now out of this part of understanding your data. So we go to the wrangling part here, which also includes a bit of transformation. And this is an aspect of the data science, data analysis that can actually take a lot of time in your project. And so having the right packages can really help you a lot and can help a lot with circumventing frustration. So for reading and importing your data, I would really recommend using the reader package. So the reader package is there to read in flat file format data. And it can be used for several formats. So it can, for instance, be used for your CSV file. But if you have a different separator, for instance, a tab delimited file, you can use TSV instead. Or you just define it yourself by using the read delim package. Note that there is a difference here in the notation with the base packages. So the reader versions of read CSV have an underscore and not a point or dot here. It also has convenient behavior that with our rectangular formats that we often deal with, that it makes column names out of the first row that it encounters. And it also has a sort of a heuristic function that sort of determines what type of data consists or is contained in a column. So it can detect whether you have your continuous double precision or your integer values, or do you have character strings. So what the description on the website also reads is that in many cases, these functions will just work. And in my experience, that is also the case. And so when you compare that then to also the base versions, they are set to be 10 times faster. So you can easily work with large data sets. And it also circumvents quite of an annoying relic from the past with the base functions, is that it does not convert your character strings to factors. So with the base version, that would be the case. With these newer reader functions, you don't have that. And you can also reverse it again, and you can write back to a file format. And in general, everything is also quite reproducible. So you get the same in as you then import out or export out of the system. So after, well, I first show also an example that's also included in the package that I created for the webinar. So if you use the function pages example that I've included here, it shows you a set of internal data files that are installed along with your package. So these are CSV files, and then you can use actually this read CSV function to see how it looks like when you import then this CSV file, so the raw file in this case. It also gives you an output where it shows you what data types are used for which columns. So the double precision here in these cases. So when we have our data, we want to get it into a tidy format. That is the philosophy that is used in tidyverse, and to get your data tidy means that you can also use the other functions more easily within this universe of packages. And the philosophy essentially states then that you have a variable in each of the columns. So that's denoted here with var. And that would be this way then. And each observation must have its own row. So that would be in this way. And then each value has its own cell. That's quite straightforward. It would seem, but I will actually show you an example where it might not be that straightforward. Now, what I also talked about already, and I will show in the exercise at the end, is that you can switch between a long format and a wide format data frame, or actually maybe the other way around, wide to long format. So when we publish data, we often have a wide format. So we have our different geochemical proxies or paleonomorph species in the column names, and the observations with a depth or height as the row. But as we have seen, for instance, for this multi-proxy faceting operation that I did, you want to have a long format data frame. So it actually sort of melts the whole data frame into a much longer data frame. And it takes then one column as a categorical that lists all the variable names, and then all the values are put on top of each other. And that's the difference. And then you can use the categorical variable for subsetting, like in the facet call that I showed before. But I will show it in action in a minute. Then we also need to be able to easily create and rename and reorder variables, and also make maybe a summarize table. And that's where dplayer comes in very handy. So I used here a couple of examples. So you can do the mutate. So this is for creating a new variable. So for instance, you can get your potassium aluminium ratio by using then your initial potassium aluminium and then just dividing it. You can select, so you can pick variables like height and potassium over aluminium. You can filter observations. So you can say, for instance, I only want to have now all observations above 3 meters, or maybe in an interval as well. You can summarize, and that's often very handy in combination with a group by call, as I showed already in the beginning. So you can group then, and then you can, for instance, group by the lithological units, so the 3 units that I showed before, and then calculate a mean value for your potassium over aluminium. So you would get a mean value for these 3 units then. You can arrange it as well. So just by ascending with height, for instance, if it is not in the right order at the first place. And then you have the group by, as I said before. So grouping according to a categorical variable, or do all the operations instead of all the column-wise operations. So everything is column-wise, do it row-wise. Can be handy in some instances as well. I include here a quite elaborate example, although I would say in terms of coding, it is actually quite lean. That I actually used also in this package to get the Bodenberg data set into a format that I wanted to. For instance, I did an oxide correction to just peel out the elements, weight percentages. And then I did also an operation that makes then the normalization with aluminium and also rename it then. And that's just to exemplify, if you have the slides within the package, then you can actually have a look at it a bit more into detail. So now let's move on to the last part. I'll have to. So what I promise you, I want to show the pivot operation, how to pivot to a long format data frame. So this would be my initial wide format data frame. And now I want to make this into a long format data frame. But there is actually a bit of a trick in this data frame. And that is that it is not tidy in the first place, because we have also information included in the variable names. Because it seems there is a grouping of these variables, like included here, like land plan. So these are one grouping, and then environment is another grouping. So we have a grouping within the column names. So we need to take care of that as well. So when we do now this elongating, so there's a pivot longer operation, we create essentially two new categorical variables and then one integer variable, which is just to count data. So we call now pivot longer, and then use our wide format data frame. I'm going to do it again in the explicit way. And then we want to take these variables, these four, and they also have the same data type. And there is actually a quite handy way to do that, because we can also say take everything except height, so that it's quite convenient, so that you don't have to type a lot. And then we say names too. So the names of the columns are going to be placed in two new columns. And as I said before, they're going to be two categorical variables, because we have additional information entrained within these column names. And I named them type and label. Well, you can argue about that, but I mean, that is how I just did it now. And then the values, so the integer values, I named them count, the new data frame, to be cost data frame. And what is now important is this last part. I say now I'm going to split these variable names based on this underscore. So in this way, it's very convenient. So we have a quite easy to recognize pattern here. So we're going to split it that way. And so when we do that, we get then all of a sudden a much longer data frame. We get then the land plan and the environment categorical variable, the label, the pollen spores, aquatic terrestrial, and then the count, all superimposed on each other. And that is also how I made this area plot with the faceting call. So it is this sequence of operations where you want to make something in a long format and then also use the faceting is something that is very convenient to know and to use to explore your data in an initial phase. And for this part, I also created a vignette, and it's also on the website, and I call it wrangling. And you can see this information again. And that's it for this part, and then I have only a recap slide later on. So Lena,
no questions yet.
No questions yet?
Nope. So I guess this is kind of the last chance if people still have questions.
You can also maybe still do it at the end. We still have a couple of minutes, huh?
Yeah.
So I just have 2 slides and then I'm done. And then any question can be asked. So to just recap what I've told now, and now in a sequence that makes, that would be the sequence that you would encounter it. In your geoscience project, you would then create first an R project. You get your organized file structure beforehand, I would say. From your data raw folder, you read your data with reader. That's the most easy way. You wrangle with it to get it into the right format. Maybe do a pivot longer to get a long format data frame. Create new variables, rename them as well to make them more easy to use within your R environment. And then you can visualize it, or you should actually visualize it first with ggplot with some basic calls based also on the data type that you have at hand, of course. You can then start to model and observe patterns. You could do that with ggplot at first with this geom_smooth call. It's very convenient. You can also use other tools to go into more detail. But as you will often encounter, it doesn't really answer your question. So you start to reiterate these steps before until you get something to your liking that is really very interesting, and then you go into more detail. So this is really the initial steps, first steps into data exploration. And then at some point you might be able to find something and you can publish and present everything. And you can also then publish along with that the documentation. So we get some reproducible science, which is a much sought after thing with current sciences crisis. Would be nice to follow this whole pathway from raw data to your process data and modeling. Last slide. No, almost last slide. So this is the data, the package again. So the data sets are included there. Raw data can be called with this function. The vignettes that I showed before, you can render the slides with render slides. And the references once more. And that's it.
You shared a lot of literature with us, but there's also a lot of cheat sheets, for instance, ggplot, right?
Yeah, that's right. So all these GitHub repos, so all these packages have their own GitHub pages, and most of these tidyverse packages also have a cheat sheet.
And GitHub is just github.com or
is it org? Can't remember. So I can show you. Yeah, this is my own .com. Yeah, can have a look at ggplot2. I'm still sharing my screen, huh? Yeah.
Yeah. Yes.
So yeah, cheat sheet is to be found in the README, for instance. But there are also other resources sites where you can find this.
Okay, so that's GitHub. Very interesting as well, next to the available books and literature. Maybe there's one final question. I don't know if it's easy to answer this one. Could you suggest ways of dealing with multiple proxy datasets for the same sediment profile?
In what way? So I guess the example that I showed, so which is also contained in the vignette and on the website. Oh no, it's only in the slides. Visualizing a multi-proxy study, if it's only, for instance, geochemical continuous data, then creating a long format data set and using a facet grid call gives you a very easy way to just have a first glance at your data, if that answers the question.
All right. Well, first of all, thank you very much, Martin, for all your work, for creating this library and giving the talk twice today at the webinar. No, it's been really great.
2 times.
Yeah. Let's see if there are some last questions. No questions yet, only thank yous.
Thank well, I would like to thank everybody as well for attending. And I hope to be of
a
help.
good sign, I guess. Yeah. All right, cool. Well, yeah, again, thank you very much. I hope you enjoyed it. I hope, thank you to everyone for joining. And yeah, like I said before, the video will be uploaded on YouTube at, well, soon. And yeah, all right.
Okay,
I guess.
Great. No, for me, it was fun as well.
That's good. That's good. Okay, then goodbye, everyone. Have a good evening, day, wherever you are. and stay healthy.